Hi guys, this is my extra credit video for Dr. Eierman's Bio202 class, and today I'm going to explain the process of meiosis with some very poor illustrations done by me. So there are some pretty big differences between mitosis and meiosis. Mitosis is cyclical, so it only goes through one cell division, and it repeatedly goes through that process, while meiosis has two divisions, and it stops after that. Mitosis is very fast and happens within an hour typically, while meiosis takes about 3 days. Mitosis occurs in somatic cells, while meiosis only happens in gametes. The difference between gametes and somatic cells is very clear. Gametes are our sex cells. In humans, they're the sperm and egg. They're also only haploid, meaning humans contain 23 chromosomes. Now, somatic cells are any other cell in your body: skin, hair, stomach, literally any other cell. Now, these are diploid and contain all 46 chromosomes. So to begin the process, we go through interphase 1. This is where DNA replicates in the cell. At this point, the cell is still diploid. Next is prophase 1, where the homologous pairs line up and create tetrads. Homologous pairs are just chromosomes that are about the same length and that typically share the same genes. While they are together, crossing over occurs. Crossing over is when homologous pairs exchange genetic material. This results in recombinant chromosomes. This exchange is part of the reason why everyone is so different. Now, I want to point out that in any other cell there would be more chromosomes, but for the sake of simplicity and my awful artwork, I just drew in one. Here's a picture to better explain crossing over because there was no way I was going to be able to draw it. Next is metaphase 1, and this is where all the homologous pairs line up in the middle of the cell. This alignment is completely random, another reason why all the cells are different. Anaphase 1 is where the homologous pairs separate. They are pulled to either side by spindle fibers. Telophase 1 is where cytokinesis occurs, and we have two separate cells. The nucleus begins to form, and now the cells are haploid. Prophase 2 is pretty simple. The nucleus starts to disappear, and the cell is ready to divide again. It's important to note that we don't go through interphase again because the DNA only replicates once in meiosis. Metaphase 2 is when the sister chromatids line up in the middle of the cell. Sister chromatids are the identical copies of a replicated chromosome, joined by a centromere. During anaphase 2, the sister chromatids are separated by the spindle fibers and brought to either side of the cell. Something that can happen in this step is called non-disjunction. Ignoring the cells I've been drawing, here are two different ones. As you can see, the sister chromatids of the purple chromosome are not pulled apart. Instead, they both go to the same side. This is a link to genetic diseases and can cause serious problems. Back to our normal cells. Telophase 2 is where the cell splits again and the nucleus forms. The cells are now completely divided and meiosis is complete. These cells move on to become sperm or eggs through different processes.